So it's my pleasure to introduce Ashley as our keynote this morning. Ashley is an alumni of the program, and we're very fond of her, and we're excited that she's here. So I'll read the intro here. Dr. Ashley Ford Versypt is an associate professor in the Department of Chemical and Biological Engineering at the University of Buffalo, where she leads the Systems Biomedicine and Pharmaceutics Laboratory. I always want to say laboratory. Her research focuses on modeling kinetics and transport processes involved in biological and chemical interactions related to physiological microenvironments and engineered biomedical and pharmaceutical systems, particularly those involved in tissue damage and treatment. Before joining the University at Buffalo in 2021, she was on the faculty at Oklahoma State University School of Chemical Engineering. Dr. Ford Versypt has garnered an impressive array of awards, including the American Institute of Chemical Engineers 35 Under 35 and the NSF CAREER. She has an outstanding record in outreach and service, chairing the American Society for Engineering Education Chemical Engineering Division in 2020 and 2021, earning an award at Oklahoma State for outstanding mentorship of women. You should follow her on Twitter because she's doing really cool stuff and she tweets about it. Not least, Dr. Ford Versypt was the 2013 Frederick A. House Scholar in Computational Science and a DOE Computational Science Graduate Fellow from 2006 to 2010. With that, let's welcome Ashley.
Thank you, Shelley, and thank you for the Krell team for inviting me to be back here today. So the House Talk 2013 was the last time I got to speak, but I have last been to the program review in 2017. So I'm going to tell you a little bit about my trajectory, a little bit about my lab, and a little bit about the outreach and other activities that I do as well. So without further ado, I won't go through all of this. The only things that weren't mentioned were that my postdoc was at MIT, my undergrad was at the University of Oklahoma, and my PhD was at the University of Illinois in Urbana-Champaign. And then I'll be showing work from my team today, and I would not be able to do this without the team, so I'll show some pictures of them throughout the talk, but very much a team effort. So this was my end of the fellowship thank you slide. When I finished in 2010 and I reused it in my House talk, but my advisors and my experience at Brookhaven were very influential to me. All of the fellows and alumni were a really big part of this experience to me as well, and then thanking DOE and Krell. These were the pictures that I had on my slide. This is coming from the left there. 2006, I was a 0-year fellow. I still had a film camera, and we explored Washington, D.C., and that's the only reasonable photo I got, but with my incoming fellow class, David Ketchison and Julianne Chung, and we explored D.C., and this is when we were still in the Washington court, so it was pretty easy to get over to the Capitol building, but we made a yearly pilgrimage to Lincoln as well. 2007, this is the cohort of other Illinois folks that I only ever saw here, but Michael Bybee was in my department, but I only ever saw him here. And then John Zuhon was an alum from Illinois who was at Chicago, but I met him and became friends with him here. Through the fellowship, I was at Brookhaven for my practicum, so I went to New York City like every weekend, which is what one does if you live in Long Island and you are not from the area. I was at Brookhaven when New York Blue opened, which was a really cool experience. I met someone at Brookhaven who lived in Beijing, and so when the Olympics happened in 2008, I had emailed her and said, hey, can I come crash at your house? And she got me tickets, and I went to the Olympics. So without the fellowship, I wouldn't have met her, and I wouldn't have gone to the Olympics in 2008. So very influential there, and then opportunities to present my science and network with people have been very influential as well. On the personal side, a cohort of Illinois alumni moved to the DC area around the time I was starting the fellowship. So I would meet up with these folks every year. And so John was a DOE fellow a few years ahead of me, but Javier and Laura moved to the area. So these are people I would hang out with every year. And then my husband Joel also is in the red shirt. So 2010 I came when I finished the fellowship. I graduated 2 years later, so I took a long time for my PhD. I did not take any pictures. I was kind of, had a little bit of imposter syndrome about how long it took me to finish my PhD. I came to the program review that year, but didn't really take many pictures. I was just kind of incognito. So I was super surprised and honored in 2013 to be the House Scholar. Like I said, I was feeling a lot of imposter syndrome, a lot of insecurity about how long it took me to finish my PhD. I like to think that I also got a degree in community service and scientific outreach as part of my PhD. I took a lot of extra classes through the fellowship and through some science education and outreach types of classes as well. And then the most recent time I was here was 2017 when I was invited to be part of a postdoc panel. And this picture is fun because it's still on the website. So anyway, so I've been happy to be part of this community, and there's lots of other things that have come out of this beyond just coming to this meeting. This is my current team, and very proud of them, and I'm very happy to be working, especially this summer with a large cohort of, I have 8 undergrads this summer, 6 PhD students, 2 postdocs, and 2 master's students. All right, so now on to the kind of fun stuff about my team. This is the first time we were all together in Buffalo. We had, with remote, some of my students moved at different times from Oklahoma, and then some of them were joining in Buffalo, but they all were there at the same time for a little bit. And then I like this bottom picture. We spelled out AICHE, which is my main professional society, so we are AICHE is kind of what I was trying to get across with that. But there's lots of fun things to do that we'll show more about. So just a little plug about my department. I've been there about 1.5 years now. Exciting to me that the strengths of the department are around computational science and engineering, material science, and bioengineering, and I do work in all of those areas. So to be in a department that's 1/3 computational only faculty in chemical engineering is extraordinary, and it's a really rich environment where we bounce ideas off of each other, where my students get lots of interesting classes. We have a separate computational data science enabled PhD program that students can also take classes to. So if you're thinking future career director, UB is a fantastic place to be for computational science. It's also a pretty location. Buffalo, Niagara is an exciting place to live and work, so many of you have probably heard of Niagara Falls and maybe even been there, and that's what I had been to before I moved there. And then this is downtown Buffalo with Lake Erie. What I didn't know was all of the kind of nature opportunities, Cherry Blossom Festival, not quite as impressive as the DC one, but still quite substantial. And then lots of opportunities for outdoor adventures on the Erie Canal. Many. This is the map of waterfalls in just Western New York. So tons of waterfalls and lots of outdoor adventure opportunities. So if you do follow me on Twitter, you will see hiking adventures, guaranteed. So Buffalo is a fun place to be. We like to do fun science there too. So the thing that we study is called multiscale systems biomedicine. And you can think about lots of different scales of spatial and temporal organization, from genetic all the way to population levels. We focus mostly on the cellular level, how different cells send out chemical signals and interact with each other, and how those populations of cells change, and how that gives rise to organ level function and whole body types of behaviors or chemical performance. And many of you have probably heard the term systems biology, and often that focuses at the molecular level or the intracellular chemical reaction network level. And we're very interested in the tissue level. Can we bridge the information that's coming out of the intracellular and pull it towards the organ level, pull it towards those things that can be clinically measured in the blood, in tissue samples, and some that are non-invasive. So we're very interested in tissue scale modeling. And so we build models mostly for 2 types of chemical engineering problems. So one of those is transport, or kinetics. The other is transport. Kinetics, so we're thinking about either biomolecules or entire cells or even entire organisms, how they react, interact, and or change their state. So on the organism side, we have some work related to bees when they're exposed to pesticides and how those bees are changing state. Most of the time, we're dealing with molecules and cells. We also look at mass transport, so we're, as engineers, very interested in how these cells and other factors move through complex biomaterials, and those biomaterials might be changing due to reactions, might be changing due to other kinds of disease states or accumulation of different cells or debris. And so we've doing these types of problems, sometimes both at the same time, sometimes one or the other, in lots of application areas. So we use a wide range of methods that are all computational. We use a lot of differential equations, both ordinary differential equations and partial differential equations. Sometimes we develop new numerical methods if they're needed, but most of the time we use off-the-shelf tools and a lot of parameter estimation in this space as well. We do a fair bit of programming/GUI development in Python and MATLAB, and those are mostly for educational purposes or collaborators who don't use a lot of coding so that we have an interface for them. We do some computational fluid mechanics and biomechanics using finite element modeling, a good bit of data analysis and dynamic systems work, and in the last 5 years, a lot of agent-based modeling. And so our differential equations are deterministic approaches. Our agent-based modeling allows us to have stochasticity and more probabilistic. These are larger, much larger scale than coarse-grained molecular dynamics, but they involve a lot of like Monte Carlo types of approaches just for much bigger physical and biomedical systems. So I'm not going to go into the details of any of these particular methods, but these are kind of behind the scenes in most of the applications we work on. In the last 8 years as faculty, we've published on a number of different types of projects. So diabetic kidney disease is a major one, cancer metastasis, immunology and immunotherapy in multiple applications, and some work on pesticides and fertilizer effects, mostly on bees. And I had a recent review last year about multiscale modeling in the human systems for diseases. So most of the things we look at now focus on fibrotic diseases. So these are diseases where there's a dysregulation in the extracellular matrix. This is the material that is around our cells and provides the structural support for our tissues. And it has different compositions in different organs, in our cartilage and our skin, for example. But extracellular matrix is ever present in our bodies and really important. And there's, when we have an injury, we'll have an immune inflammation, we'll have cells that are signaled to try to heal that wound and form some fibers, but that can get dysregulated where there's more scarring or chronic fibrosis that results. And so we're very interested in those processes that lead toward healthy healing or dysregulated, and then also some processes like osteoporosis that break apart fibers and that we want to try to get them back towards a more fibrogenic phenotype. And so the fibrosis exists in many different organs and disease states. And so we, as a team, look at several different physiological systems, and we think about them on the whole organ level, but sometimes we have subcompartments of the organs that we're looking at. So right now we have active projects in the bone, the lung, kidney, eye, and cancer, specifically breast cancer tumors. And first I'll tell you just a little vignette about the kidney part. So this is a long-standing team effort. This is work that I started while I was a postdoc independently with some math faculty, a group of other women actually in mathematical biology. And so those are the earliest publications there in 2015. And then with undergraduates and graduate students in my lab across the last several years. So this is, I believe, all of the students on my team who've worked on this project specifically over the last 8 years. So quite a number of folks. We're looking at a certain zone of the kidney that is damaged due to diabetes that changes how our blood is filtered, and it involves some cell death, some different dysregulated chemical processes, fibrosis, et cetera. And this is work that's supported by the NSF CAREER Award. So this is just a zoomed in shot of this area in the kidney. What you're seeing in this kind of circular or elongated circles is cross sections of blood vessels. So we have these clusters of capillaries where our blood is filtered, and normally the waste will be exiting, kind of following the paths of those arrows, and being collected to form urine in this outer sort of surrounding zone. In diabetic kidney disease, a number of different perturbations happen where the vessels can get squeezed because more cells and fibrosis, more cells grow and fibrosis happens. So in the same volume, we have more stuff, and so everything is more compressed. We can also have cells on the outside, the yellow ones, that die off, and that's deleterious. And so we are modeling different subproblems happening in those blue boxes. I'm not going to go into all the details of that today. What I wanted to show you involves our outreach efforts from this. But in a summary, we're working on connecting the biochemical networks in those different cells, the transport processes between those different cells in the specific environment, and this tissue structure crosstalk with a stochastic agent-based approach. So we're putting together disparate pieces and building our multiscale model there. So our outreach efforts and our broader impacts involve substantial efforts of undergraduate research. So I have the universities the students are affiliated with. It's no surprise that there would be Oklahoma State and Buffalo students, as those are my two institutional affiliations. But I've also had students from Emory University, Howard University, and Case Western Reserve University who've been part of the team during summers. And students are from chemical engineering, mechanical, biosystems, biology, math, microbiology, biomedical engineering, and nursing. So, and they're all doing computational work. And so it's been really fun to work with them and to onboard them into this kind of work. And so we use mathematical biology and differential equations as an onboarding tool for these students as they go on to work in my lab. We also have, as part of the career, integrating teaching, research, and outreach through a number of different things. So one of them is a Women in Science Expo. So the state of Oklahoma had an event annually with about 1,500 middle school and high school girls, and we would come with the GUIs that we had made and set up a table and have women from my lab and men from my lab staff the booth and have interactive activities with our programs. We also worked with what was called the Summer Bridge program, which was for incoming College of Engineering freshmen. We're going to be taking the same program and using it at Chemi summer camp in 2 weeks in Buffalo. We also had an event at Oklahoma State called Grandparent University, which is where students and middle school students and their grandparents could come to campus for a 3-day camp, and they picked a major, and they studied that major for 3 days, and they did activities, and they slept in the dorms, and went swimming, and watched a movie on campus, and a lot of other fun activities, met the mascot, etc. So this was our activity of them being a molecule in a pore, and we were talking about filtration. And so they hop on one foot through channels of different widths, so one is very narrow, and they're trying not to touch the tape. And so here it's very easy. They're a small foot in a big channel. The next one to the left is a narrow channel and the same size foot, so they're bumping it. And then we do another channel that we make double-sided tape, and they're sticky, so trying to demonstrate adsorption to the surface for more of a chemical separation. And so they hit that tape and they get stuck to it, unlike when it's single-sided tape. And so they have a very physical reaction to, oh, wait, I'm stuck to this tape, and we use that to discuss mechanical-based, size-based separation and more chemical adsorption-based separation techniques. We also have done things at this local kids science museum, courses. I have a numerical methods course that was kind of tailored after software carpentry, but intended for seniors and graduate students across campus to introduce them to scientific computing, technical writing with LaTeX, MATLAB, and Python for not basic skills, but for being able to actually use the algorithms that are in MATLAB and Python. So you're not doing like a forward Euler detailed code, but you're using the better packages for better ODE solvers and other kinds of things. And so that course is packaged on GitHub and distributed for, on my page, which is github.com/AshleyFV. It's APPL num comp for applied numerical computing. We disseminate our work through ASEE and chemical engineering education papers. And then next week, we're presenting at the Chemi Summer School for other engineering faculty. So it's a week-long camp, essentially, for early career chemical engineering faculty. And my collaborators, Matt Stubert and Robert Hesketh from Connecticut and Rowan University, were putting together a set of essentially MATLAB live scripts and Jupyter notebooks for teaching linear, nonlinear equation solving, ODEs, boundary value problems, and PDEs across the Chemi curriculum to faculty who may not touch computers. And so for them to be able to integrate these into their courses pretty regularly. And so we're going to teach them about how to make basically a Jupyter notebook. So that's next week. So those materials will be shared, but they're going live next week. So I tell you the career story because of the kind of educational portions of it. The most recent publications coming out of my lab are actually from our bone collaboration. And this has been very exciting for me because this is our first funded collaborative project. My earlier grants were all single PI, which is good, but I like the collaborative science a lot. This has been really fun and rewarding. So on the bone project, my main collaborator is Brenda Smith, who is a nutritionist who also is an expert in bone health and uses mouse models to test nutritional supplements' effects on the bone. And she was in nutrition at Oklahoma State University and has recently moved to obstetrics and gynecology and musculoskeletal health at Indiana University. And then on my team, Mohammed Ahmed al-Islam is a postdoc, and Carly Cook was a master's student, now a PhD student in my lab, and who've been the primary drivers on this work. So the first paper from this work was featured in Industrial and Engineering Chemistry Research in December in a special issue called Influential Researchers. And then just a couple weeks ago, our second paper on this was published in the AICHE journal. Both of these were invited, and they were key publications in the chemical engineering space. And so at this stage of my career, I need to have visibility in the chemical engineering space. This work, the collection of this work, I'm invited to speak on it at the Foundations of Systems Biology and Engineering Conference next month as a keynote, so I'm very excited about that. And then because of the AICHE journal paper, it was invited for a special session at the upcoming AICHE conference. So again, just fun work coming out of this bone project for us. So you will get to kind of see the first draft of this presentation about this work. So what we're doing behind the scenes, I don't really have much of the math part in here, but we are building mathematical models using differential equations and doing a lot of parameter estimations specifically to, matching to this biological data. So we're looking at what we call the gut-bone axis. How does the gut connect to the bone? And the implications of butyrate treatment. So butyrate is a short-chain fatty acid that is fermented in the gut, and you can give it directly, or you can give fruits and other fiber sources that cause the gut microbiome to produce more butyrate, and that's what we're most interested in. In this particular work, we were looking at data from giving the gut new bacteria to enhance the production of butyrate, and we've seen that that can enhance the immune suppressive environment to reduce inflammation. And inflammation has a role in promoting the degradation of the bone. And so we're interested in suppressing that information and allowing the bone to build back up. So we built a PKPD, that is a pharmacokinetics, pharmacodynamic model about how the butyrate dose is affected to the impacts both the distribution throughout the gut, the blood, and the bone, and the effects on the body. That's the pharmacodynamic part. So we also coupled this with, I mentioned the butyrate in the immune cells, they have an impact on the bone health. We couple this to what's actually happening in the bone with detailed cellular model there. So I'll show a better, more zoomed in version of that image on the left in a few minutes. So gut connected to this immune system and then to this detailed model of the bone compartment. So there's a lot of data here, but this is a study published in 2018 where they gave mice new bacteria to enhance butyrate production. And so the LGG is the new bacteria, and the butyrate, you can see, goes up in the small intestine and in the serum, so that's important. These other small short-chain fatty acids did not go up, so it was butyrate in particular. And these are different types, or this is all in different zones of the body, a certain type of these immune cells, these T regulatory cells. So they showed that went up. So they knew Tregs are involved, but this is data about the skeletal response. So the interesting part is that they saw really good improvement of the bone volume through this process, and we wanted to be able to capture that. And we also wanted to be able to capture some of the effects of this Wnt10b, which I'll talk about more in a moment. So my postdoc, Aminal, built this multi-compartment model that could connect from the butyrate in the gut and the immune cells in each of these three zones, how they moved around, how they promoted. It is believed that the butyrate causes naive cells to become T regulatory cells through differentiation. So that's the main biochemical process we included. And then once in the bone, there's a cascade of chemical effects that enhance Wnt10b. Then from that TGF-beta and Wnt10b, we then connect it later to our detailed bone compartment model, which is our second paper. So we could connect this, or we used data to parameterize some parts of the model, and then we validated against other parts of the model, against different data that weren't used for validation. So we have consistent performance in each of those three compartments, both with data we fit to and data we used for validation. We also calculated, so this was the kind of the positive case. You give more butyrate through, or you get more butyrate through having this dose of the new microbes. This was an antibiotic treatment, so lowering that butyrate. We also got good agreement in our full model. Then the Wnt was the part that we really needed from that first model. So given some sort of butyrate change, what's the corresponding Wnt change dynamically? And so this is both for the adding the new amount of microbiota, and antibiotic treatment is in the orange. So we're going to use that later in connecting to our other bone compartment model as an input. So this is from that paper that had the original data for this probiotic treatment about their working model of the mechanism. But in biology, a working model is often just a diagram. There's no math behind this. There's no testing whether that, there's testing on the animal if it's still consistent, but not quantifying the dynamics of this. And so we sought to build a model based on this working model from the experimental folks. So from the gut, having the butyrate influence these CD4 plus T cells, those are our and have them become Tregs, have those enhance the amount of Wnt10b, and then for that to have an effect on the bone compartment. So that first part of the model was about how the Wnt could get produced, and then I'll tell you more about how that influences the bone compartment. So this is the bone remodeling cycle. So in our bodies during normal conditions, our bone is being reformed. You might experience some mechanical stimulation or some sort of other chemical factors that starts sections of your bone into remodeling. And so small sections undergoing this remodeling process very frequently. And so let's start from this activation at 9 o'clock, if you will, on this circle. So there's some stimulus that happens where an osteocyte that's in the bone tissue has been damaged, and that triggers this remodeling cycle. So pre-osteoclasts, the purple ones, will there's a pool of them already there. They will be signaled to proliferate and differentiate and make new osteoclasts. These are the ones that clear out the existing bone tissue locally, and then the pre-osteoblasts are the ones that are going to regulate that and build back bone. So they stop the process of the osteoclasts, and they come in and build back in new bone matrix, and then it gets mineralized And some of those osteoblasts are deposited into the tissue as osteocytes that then can start this process again later. So this is the foundation that we use in understanding for building a model of this process with these key cell types. So if you have osteoporosis or some other bone damaging disease, the cycle of too much osteoclasts breaking apart the bone is out of balance and you have a net bone loss. If it tips the other direction, where there's too much osteoblast, you can have too much bone formation. Most of the time, the body's going to stop when it actually hits a reasonable bone volume and not accumulate too much bone. And we're mostly interested in recovering from an osteoporotic bone loss phenotype to get back towards a healthy condition. So this is our starting point. So here, the circles are representing different of those bone cell types. The preosteoclasts are the PC, the P is the preosteoblast, B is blast, the builders that are leading to more production of bone, C is the osteoclasts that are cleaving, breaking apart the bone, and the S is the osteocytes. So there is an existing model amongst several models, and we evaluated all of the suite of models, mathematical models that already existed for bone homeostasis. So in healthy conditions, how is this remodeling process happening and which things drive net bone formation? So we used work from Graham et al., but again, surveying several from multiple journals. And it has power law relationships for different chemical factors. And they had a cycle length of 100 days. We sought to add, you see there's no chemicals on any of those arrows, so we sought to add the impact of Wnt10B on this. So we altered it beyond one remodeling cycle because we wanted to connect to this data that had different time periods, and we wanted to have, what if we were chronically giving Wnt or stimulating Wnt through the different gut, either bacteria or through more butyrate, or food sources of butyrate. So we have 3 places we added this Wnt dependence: directly on the proliferation of those cells labeled P, on the differentiation from P becoming B, and slowing the death rate of B. These were informed by the literature from experimental data. We used Hill function form, so it was not a linear, but a saturating effect. And so Wnt has been shown to increase the proliferation. So there was already a proliferation term, but we enhanced it by this factor pi from the Hill function. We also then increased the osteoblastogenesis and slowed the apoptosis. We put those into the model, so we had 4 new parameters that we needed to estimate. We only have data for kind of the end of some of these cycles, not intermediate cycles. And so if we had more data, we can improve this model. But this is showing where the data are and where our simulations are, and that we used for our parameter estimation. So these were data sets from the same lab using the same techniques for different amounts of time in different amounts of Wnt that were exposed to these animals. And then this was separate data we used for validation from a different lab. And so particularly for the 6 cycles, we had really good agreement. For the longer, it started to deviate, and we probably need our model to taper off rather than continue to grow. But that was good for now. It's giving us preliminary data as we're putting in other proposals to try to fund having a more time course related animal study, which is very expensive to do. So we can predict all of those different cell types that I talked about, how their dynamics happens. What you should note from this is there's not much change peak to peak. So there are cycles that are happening, but there's not much variation cycle to cycle. Here is showing different doses of that Wnt as an input to this bone compartment model, and how that does change the populations of some of the cell types. So the osteocytes, we manually lower to 180 at the beginning of each cycle. So that one responds the same way every time. The preosteoblasts are increasing a little bit with the amount of Wnt that is dosed. and increasing the osteoblasts more significantly and decreasing the osteoclasts. But these are small numbers on the osteoclasts. The osteoblasts are much larger populations. So this was useful for us in like verifying against some studies that were already out there. The more interesting part for us was being able to connect this to the full pharmacokinetic model from the gut all the way into the bone. So we have that Wnt as the output from the first model that becomes the input to the second part. And so we did connect these, and this was the simulation result for with no butyrate dose, the Wnt is just at its baseline, and then with the positive dose. And what we see is that when we connect the model, we get just a little bit of response in the bone volume, but you see this data point up here? This is the measurement. So there's a big gap here. And so we thought this presupposes that the only way that the butyrate is affecting the bone is through this intricate network of processes. It triggers something at the beginning, the immune system takes over, and it's just this cascade of effects. But this led us to believe that that small amount of Wnt change wasn't actually enough to make these substantial bone changes alone. It contributed, but it wasn't sufficient. And so then we conjectured that potentially butyrate may have a direct role. So instead of butyrate having to go through this complex pathway, it still may do this, but butyrate may directly enhance some of these processes in the bone. So we sought to test that mathematically. And so we did a number of different cases that I won't go into all the mathematics of, but basically one term at a time, we added a direct dependence of butyrate in our model and tested those one at a time to just match them up with this data point after 2 cycles and saw what could any of these be explanatory. And so they all can get to that data point, but then we explored their actual bone cell responses. So some of them, of those cases 3 through 9, gave non-physical types of performance of the different individual cell types. So we rejected the ones that cause osteoblasts to decrease, that's not consistent. And we rejected a couple others that were just like, butyrate makes bone enhance without any of the mechanistic details. So then it gave us really 3 accepted mathematical hypotheses that then now we're designing new experiments to test with our collaborator in their animal models to determine if butyrate is directly having impact on the proliferation of the osteoblasts, the differentiation of osteoclasts, or the apoptosis of that osteoclast. So for me, this has been a long time coming that I've used a lot of secondary data in the past. My entire career, this is the first time that we're working directly with someone to plan the experiments and that our models can inform the design of new experiments. So it's a really fun collaborative environment to be working in now. So we've gotten some new insights on the osteoimmunology. We've been able to add this mathematical term for kind of the saturation of the Wnt effects. We've been able to see how Wnt influences the bone compartment, but then also generate these hypotheses for direct roles. So I haven't told you about all of these other cool environments that we study, but they are there, and we have some publications yet. We don't have any publications on the eye, but we did have a really positively reviewed R01, so maybe that'll get funded, and then you might see some work coming out from us about the eye work in the next year or so. So with that, I think that's the end of what I have. This is our Buffalo History Museum during the Cherry Blossom Festival. I love cherry blossoms in my lab here. I'd love to connect with any of you, be happy to take questions about any of these kind of activities I've mentioned, both the scientific, mathematical sides, my career, our outreach activities, Buffalo, life in general. So I'm very happy to be here, and thank you for the opportunity to present today.